KURINJI Senior Secondary School, CBSE, Namakkal.
Successful result producing school in state every year.
Administered by the Board of Directors with over 35 years of experience in the teaching field.
Special attention on art, craft, music, dance, silambam, yoga, skating and karate.
Focus on Hindi Sabha and Spell Bee Olympiad exams.
Smart class for effective and innovative learning and well-furnished library.
Solitary importance on sports and games in state and national level.
WhatsApp facilities to know activities of students and school inside the campus.
NEET, JEE, IIT coaching experts from Rajasthan and Andhra Pradesh along with our effective teachers.
Admission registration for classes LKG to +2.
KURINJI Educational Institution, Kavetipatti, Namakkal. Admission in progress. CBSE, KG to 12, contact 9025895176. Metric 6 to 12, contact 9344567484. For online admission, log on to www.kurinjischoolnkl.in.
Students, now in this video, just we are continuing the information of nitrogen cycle. Okay, so in previous video also we have given half of the information. Most of the information we have given. So now previous video continuation, you try to watch this video. Now nitrate, in previous video one more time. In previous video we see know what it is. So with the help of nitrosomonas or nitrococcus, so ammonia is converted into nitrites, later nitrates. So nitrates later converted into nitrates. So in the presence of nitrobacter, likewise we see now, so means up to the formation of nitrates. So we have stopped the previous video. Now continuation of the video is the nitrate thus formed is absorbed by the plants and is transported to the leaves. So means what is a nitrogen form, means nitrate is a form, no, that particular nitrate is absorbed by the plants, absorb the plant and transported to the leaves. Okay, now in leaves what is happening means, in leaves it is reduced to form ammonia. Point you understood, no? In leaves again it is reduced to form ammonia, means so thereby point you understood, no? Actually ammonia is converted into nitrates to nitrates form ஆயிடுச்சு, அப்புறம் nitrates to again what that has been entered into the plant system and finally reached to the leaf. Again in the leaves, no, in the leaves the reduced form again ammonia formation takes place. That's finally forms the amino group of amino acids. Means one more time, ammonia is the ammonia nitrate madre change nitrite later nitrate to it enters into the plants and in the plants or the leaf of the plants, no, again it is converted into ammonia. So why here the ammonia has been formed means, so this particular ammonia helps to form the amino group. So amino group of amino acids means for the formation of amino acids, amino group is formed from the ammonia. So this is the point. Now nitrates present in the soil is also reduced to nitrogen by the process of denitrification. Means here when the process is going to be taken place like ammonia to nitrite to nitrate, if nitrate form is there in the soil, no, at the time there some kind of denitrifying bacteria also are present in the soil. At the time denitrifying bacteria, what it will do is, so what are the nitrate compounds are there, no, or the nitrate form of nitrogen. So this one will be again some or very few or the little quantities of the nitrogen or the nitrate, again it will be reduced and enters into the atmospheric conditions. So because of the activity of denitrifying bacteria, and the process is known as denitrification. Denitrification is carried out by Pseudomonas and Theobacillus. Pseudomonas and Theobacillus, what it will do? Generally nitrogen fixing bacteria fix the nitrogen in the soil, but Pseudomonas and Theobacillus bacteria, what it will do is, so reverse work. Reverse work means from the soil again this role do the some kind of denitrification processes, and nitrogen forms again because of these two activities. Again it will be converted into atmospheric nitrogen. Actually most of the bacteria what they are doing, so what is the nitrogen is present in the atmosphere, no, that is converted in and converted and forms the some kind of forms and keep the nitrogen fixation or do the nitrogen fixation in the soil. But some other kind of nitrogen fixing denitrification bacteria, what they will do is reversely they will act. Means what is the nitrogen is present in the soil, so by following of denitrification processes and all, try to reduce the content of nitrogen in the soil. So that particular point only we are taking as denitrification. Now the denitrification is carried out by the bacteria is Pseudomonas and Theobacillus. Now Pseudomonas and Theobacillus bacteria, so which plays an important role in the process of denitrification. Later, biological nitrogen fixation. Biological nitrogen fixation. Now here, very few living organisms can utilize the nitrogen in the form of nitrate, in the form of N2. Or one more time you see there, very few living organisms can utilize the nitrogen in the form of N2 available abundantly in the air. So thereby here what is happening? So only certain prokaryotic species are capable of fixing nitrogen. So means here abundant quantities of the nitrogen is present in the air, but very few kind of living organisms only can utilize the atmospheric nitrogen, so which is there abundantly in the air. But only certain prokaryotic species are capable of fixing nitrogen fixation. Now, reduction of nitrogen. The reduction of nitrogen to the ammonia by the living organisms is known as biological nitrogen fixation. Now, here you see there the reduction of nitrogen to the ammonia means here the reduction of nitrogen to ammonia formation, it is taking place by the presence of living organisms is known as biological nitrogen fixation. Means here when the nitrogen process or the nitrogen fixing process is going on, there is a involvement of living organisms. That's why we are taking the biological nitrogen fixation. Next one, the enzyme nitrogenase. The enzyme nitrogenase, which is capable of nitrogen reduction. Nitrogen reduction means it's nothing but it is a kind of ammonium preparation. So thereby our knowledge point of view, oxidation and reduction means reduction is nothing but addition of hydrogens or the removal of oxygens, we will see now. So such kind of a process are going to be taken place. So entire this process when it is going to be taken place, it mediated by the enzyme known as nitrogenase. So now this particular process it's taking place exclusively in prokaryotes. So such particular prokaryotes or the microbes, we are calling them as nitrogen fixers. Nitrogen fixers means living organisms are there. All kind of living organisms cannot utilize the atmospheric nitrogen at the time. No, few of the living organisms are having the capability to utilize the atmospheric nitrogen directly. Okay, so later what is happening? So what are the microbes are having of nitrogenase enzyme? So they have the capability to fix the atmospheric nitrogen, so in the form of ammonia. So such kind of microorganisms only we are calling as nitrogen fixers. So the process is known as biological nitrogen fixation. Now N triple bond N. This is atmospheric form only, no? In previous video we seen. Strong covalent bonds are present, 3 we see, no? Adida. Now with the help of nitrogenase enzyme, which one formation taking place here? NH3 formation means ammonia formation is completed. Now the nitrogen fixing microbes could be free living or symbiotic. So now here we may get one doubt, sir, here there are different types of microbes are there, no? So how they will be, or how they will be involved in the process of nitrogen fixation, either free or symbiotic mode means, so both chances are there, or both types are there. So now examples of a free living, examples of free living nitrogen fixing aerobic microbes are Acetobacter and Bicernechia. Examples of free living nitrogen fixing aerobic microbes. Free living nitrogen fixing aerobic means, so in the presence of oxygen also definitely, aerobic microbes, so they can work. So such kind of microbes examples, one is Acetobacter and second one is Bicernechia. And while Rhodospirillum is anaerobic. Path these examples, NEET examination point of view important. Acetobacter and Bicernechia are aerobic microbes and free living. So free living and aerobic. And while Rhodospirillum anaerobic. Okay. And the bacillus also is free living. And in addition, a number of cyanobacteria such as Anabina and Nostoc also are free living nitrogen fixers. Means here bacteria's point of view, there are two different types of bacteria we have seen. So one is a free living we seen, as well as and another type is symbiotic also we see. So some kind of a bacteria have the capability so to fix the atmospheric nitrogen even in free living conditions also, means no need to depends on the any other kind of organism as a symbiont. So by being freely also they can fix the atmospheric nitrogen. So already we have seen Acetobacter and Bicernechia are free living aerobic microbes. And free living anaerobic Rhodospirillum comes, and Bacillus also comes under free living one. In addition, a number of cyanobacteria, even a number of cyanobacteria such as Anabina, Nostoc also reported as a free living nitrogen fixers. In cyanobacteria means here exclusively we are going to discuss the information related to the bacteria as well as cyanobacteria. In bacteria we have seen what it is, a free living one, aerobic, anaerobic. Likewise in cyanobacteria now we are seeing the free living nitrogen fixers. Examples are Anabina and Nostoc. Okay, so that is the information. Now symbiotic nitrogen fixation. One more is symbiotic biological nitrogen fixation. So how it will be? Symbiotic biological nitrogen fixation. So means here free living is there and symbiotic is there, we see no. So there symbiotic one we are discussing, the symbiotic biological nitrogen fixation. Now several types of symbiotic biological nitrogen fixing associations are known. Means here there are different types, or there are several types of biological nitrogen fixing associations are there. So but the most prominent among them is the legume bacteria relationship. So means here when we are going to start the biological studies, no, so means in 8th standard or 9th standard or in a 10th standard definitely, so chance is there to study the relationship between the legume plants with the bacteria. So this is the most prominent one. Means this one, it is a very familiar, this particular relationship maximum all biology students ku theriyum. Okay, so that is the information now we are going to discuss in a deep manner. Now the species of rod-shaped rhizobium has a such relationship with the roots of several legume plants. One more time. Suppose if you see the species of rod-shaped rhizobium, so rod-shaped structure rhizobium means here one point whether you remembered or not. Based on the shape of the bacteria, 4 types of bacteria are there, no? Cocci, bacilli, vibrio, spirillum. Likewise, cocci is spherical shaped, bacilli is rod shaped, no? So thereby here even rhizobium bacteria also comes under the bacillus type. Okay, so thereby here what is the thing? The rhizobium bacteria has the relationship with the roots of several legume plants. Roots of several legume plants known as alfalfa. Okay ma'am, now you see there what are the plants here, the several legumes are there means alfalfa, sweet clover, sweet pea, lentils, garden pea and broad beans, clover beans. Likewise there are different types of plants or the legume plants we can be seen. Even examples are important, so panikunga. One is alfalfa, sweet clover, sweet pea, lentils, garden pea, broad bean and clover bean, etc. etc. not written but here etc. Still so many are there. So all these are all we will be taking them as several leguminous plants. So definitely leguminous plants means definitely they will be associated with the rhizobium bacteria. So by associating with the some kind of nitrogen fixing bacteria, so definitely they will be benefited with the nitrogen quantities. So why? Because suppose if the nitrogen availability is there means, so that one they will use for their metabolic activities. So that is the information. Later the most common association on the roots is as nodules. So means here the most common association on roots is as nodules. Means here first of all what is happening? So when the rhizobium bacteria is enters into the body of the plant root system, at the time it forms the nodules. Means over like spherical shaped structures, small spherical shaped structures formation takes place. So these spherical shaped bulged structures only our subject point of view we are calling as nodules. So now these nodules are small outgrowths of the roots. Now we see these nodules are small outgrowths of the roots. Now the microbe and frankia is also produces the microbe. Even this point also important, nalla save pannikonga. The microbe frankia also produces nitrogen fixing nodules on the roots of non-leguminous plants. Example alnus. Means here non-leguminous plant example alnus and microbe example is frankia. Microbe example frankia. So what it is doing is it's a produces, means here the microbe of frankia, it is associated with the root system of non-leguminous plants alnus. Okay, now both rhizobium bacteria as well as frankia are free living in the soil. So free living in the soil, but as symbionts can fix atmospheric nitrogen. So means early rhizobium bacteria as well as frankia both are free living in the soil. When these two are free living in the soil, at the time not having the capability to fix the atmospheric nitrogen. But when the rhizobium bacteria and frankia are attached to the root system of leguminous plants and non-leguminous plants respectively, that time only they can fix the atmospheric nitrogen. Rhizobium bacteria will go and binds to the root system of leguminous plants, and the frankia will go and binds to the roots of non-leguminous plants. Okay, at the time definitely both can fix the atmospheric nitrogen. Okay, so thereby here this is a point to be highlighted. So konjam understanding pannikonga. Appuram uproot any one plant of common pulses. So just before the flowering. Suppose if you notice, suppose if you observe, suppose if you plug the one plant and if you observe there uproot plant, so definitely means here any one of the plants we should not take, means here we need to take care about the pulses. So any one of the pulses plant, suppose if you uproot and if you observe there before flowering, definitely what we can observe means you will see near spherical outgrowths on the roots. Means here the spherical shaped structures we will notice on the roots. Now these are nodes. Means what are the spherical shaped structures means here the patham both. So in this particular diagram also the nodes are available, you can watch. So thereby these spherical shaped structures we are calling as nodules. Now if you cut through them, so you will notice the central portion is in a red color or pink color. So here, so if you see or if you cut the central portion, suppose this is a nodule, for example, if you cut this one, if you cut the nodule with the central portion, no, so definitely we will find out or we will notice the red color or pink color pigment. Sir, why sir within the nodule, why sir red color or pink color is there? Means this is due to the presence of leguminous hemoglobin. Leguminous hemoglobin or leg hemoglobin. Okay, so thereby means here the red color or the pink color pigment which is present in the legume or the nodule, so which protects the or it is protects the rhizobium bacteria. Why? Because here the rhizobium bacteria, when it is in a free living, no, it survives in the presence of oxygen, but when it is acting as a symbiont, no, or the symbionts, no, at the time definitely it is act as anaerobic bacteria. Anaerobic bacteria. So thereby anaerobic bacteria means here which one is not required? Oxygen is not required for their surveillance. So that's why here, that's why this rhizobium bacteria which is protected from the or protected by the leg hemoglobin. Leg hemoglobin, the leg hemoglobin color is red or pink. Okay, so means here the what is the point we are discussing? The leguminous plants hemoglobin is there, that one we are calling by hemoglobin or leguminous hemoglobin or leg hemoglobin we are calling. The what is the work of this leg hemoglobin means it acts as oxygen scavenger. This new point save pannikonga. It acts as oxygen scavenger. So means oxygen scavenger means, so what are the in that particular atmospheric conditions, suppose if oxygen levels are there means, so that particular oxygen is removed by the leg hemoglobin. So thereby in that particular locations, no, there will not be the presence of oxygen. So thereby anaerobic conditions will be created. So at the time rhizobium bacteria survives happily. Okay, so thereby here, so here the rhizobium bacteria by protecting the leg hemoglobin, so fixes the atmospheric nitrogen and helps to the plant body. So likewise here in the soil, so gradually or the year by year definitely we will notice the fixation of atmospheric nitrogen by the leguminous plants. So this is the entire information related to the symbiotic biological nitrogen fixation. In the next video, we will have the information how the nodule formation taking place. So how the nodule formation taking place in the leguminous plants, very exclusive information we will discuss. But they watch this video clearly and gain the knowledge and be safe at home.